We are all meeting here today in this virtual event under very unique circumstances. I wish we could be meeting in person, but right now, the first and foremost thing that I would like to hope for is that all of you are doing well and staying safe. As someone who grew up in India and still has a family all over India, I am very concerned about the current pandemic rise in the country, and I truly hope that things get under control very soon. Now, as Lakshmi said, I come to you from Arizona, and even though the sun is not out yet, we are guaranteed a bright, sunny day today. And as someone who enjoys these bright, sunny days, I'm especially delighted to be part of this kickoff for the Inai Center for Population Scale AI, because I truly believe that in collaboration with the government of India and Indian academia, this center is going to be that bright, shiny spot of hope in overcoming many of the challenges that we face today. So if you truly look at this pandemic, it is one of the ones in our lifetime that has been truly global in scale. And while rightfully all the governments are really trying to and scrambling to provide for immediate healthcare as well as economic support for their people, one other reality has set in the minds and worlds of science, which says that we are going to have to look at some of the technological capabilities and services that just a few years ago we thought were aspirational. They are now going to become essential. Things like biomarker prediction or disease subtyping, gene therapy, drug discovery, all these things that were something that we thought we would get to someday are now going to be critical as we prepare ourselves for some of the cures and therapies for not just COVID-19, but the next pandemic that will definitely rise. So this is where genomics and genome sequencing is becoming one of the foundational pillars as we look for these therapeutics. Now, I will upfront admit that I'm not a biologist, and I'm not going to turn this into a session on genomics. But let's just say that genes that are made up of DNA essentially control, they determine how various proteins get developed in our bodies, and proteins are the ones that determine how the cells in our body behave. And there is a whole process of transcription, translation, folding, etc., that happens within our body to go from these DNA base pairs to the ultimate proteins that, as I said, control how our bodies behave. And the whole phenomenon of genome sequencing, what it requires is that you have to take DNA molecules and break them down into small molecules, then read them back, and then put them back together to create this genome sequence for an individual. And the way you do that is you kind of use a reference genome, and as you are building this sequence, you compare it to this reference genome, sequence, and you look for what we call deviations from that reference, and that are called the variants. And this study of these variants really provides clues for you to really figure out what are some of the genes that might be contributing to some of the diseases and maladies in our bodies. And then that leads to the study of what kind of proteins might be causing these problems. And then you come up with the right therapies or drugs to really disable those rogue proteins in our body. So this whole process of genome sequencing, if you look at the rise of genome sequencing, as the chart on the left-hand side shows here, that processing of genome has been doubling every 7 months, as shown by the red line over there. Now, everybody knows the Moore's law, which says that our ability to pack compute capacity into our platforms doubles roughly about every 18 months, which is the blue line. And if you look at the estimate from Illumina, one of the pioneers in this field, they may say that it may not be the red line, but maybe the yellow line is kind of the requirement in terms of how quickly genome sequence is going to rise on the planet. So clearly there is a gap in terms of what the underlying infrastructure can provide and what genome sequencing would require. Now, if you look at the chart on the right-hand side, what you see is that publications involving use of AI for this field of genomics has been just exploding. So it is very clear that AI, with access to large data sets, and AI requires large data sets for it to be more effective, it is going to come to the rescue as we try to tackle this whole field of genomics. And some people estimate that by 2025, we would have about 2 billion of human genomes sequenced. But you recognize that that is still probably at that time only roughly a quarter of the world population. Now, this is just the field of genomics, right? So if you look at what kind of underlying processing infrastructure you would require, what we will need is almost 1000x of compute capability. And then it will require hundreds of terabytes of data. Somehow I lost my screen. Just give me 1 second. Okay. So we will require almost 1000x of compute power and significantly large amount of storage to deal with the data that we are going to have to deal with. And at the same time, one of the challenges for this is going to be that since we are dealing with people's data, people's very, very confidential data, we will have to make sure that security is also going to be a key component of the solution that we come up with. And by the way, this is just the field of genomics. You add this to other type of field of science, which is like proteomics or microbiomics, etc. And you basically end up with hundreds of exabytes of multimodal data that we will have to process to deal with this dynamic. So what I submit here is that we cannot just throw more and more compute power at these solutions. What we will need is a systems approach where you will have these huge cluster of compute nodes, as well as you add the storage nodes to it and the networking infrastructure that connects these compute nodes and storage nodes and moves the data around between all these different nodes. And we need to have a balanced systems approach if we are to really meet the requirements of what is going to be required from that infrastructure to do this kind of omics processing, as we call it. And then on top of it, as I said, we need to overlay the security piece to make sure that people's confidential data is preserved. So now, if you look at what we have been doing within Intel and Intel Labs, we have been working to enhance the compute performance for many of these AI workloads. And as you can see, just in last 2.5 years, we have been able to improve the AI inference performance using our Xeon platforms by almost 1500x. Also, if you look at, and by the way, it is not just underlying hardware that improves, we are also making significant improvements in the algorithms that people use to process all this data. As you can see on the right-hand side, the Burroughs-Wheeler aligner is one of these algorithms. I mentioned earlier that the way you do genome sequences is that you break down the DNA molecules in smaller pairs, and then you kind of put those pairs back in a particular sequence. And for that, this BWA algorithm is critical. And we have been optimizing that algorithm to run on our platforms significantly. And as you can see, we will continue to do those kind of performance improvements for all these AI algorithms. As I said, it is not just about compute. We also have been working with the world-leading Broad Institute to develop this Genomics DB, which is essentially a data store that is optimized for genomics data. And it is built on top of this another technology called TileDB, which we developed to make sure that data is organized in a fashion so that as you are trying to read this data, it is accessible in a very easy fashion so that the algorithms can process that data more readily. And as you can see here, we have improved the time it takes to query a lot of these different types of genomics data. And as you can see, we have brought the latency significantly down as you increase the number of samples for this. And in fact, we got this quote from one of the leading researchers at Broad Institute that said that the processing time for some of these variant discovery went from 8 days to 18 hours for 100 whole genomes. And to them, this was a breakthrough that was truly transformative. And we will continue to do these kind of innovations. And by the way, we also believe in open source and high performance platforms. So that is what this NI Center also embraces, and we are really excited about that. Similarly, we have brought a lot of performance improvements in the networking subsystem. And when you try to pull all these pieces together, and I'm getting my cue for wrapping up, what we have to do is we have to go from this single omics to integrated omics, where we bring not just the genomics, but proteomics and all these other different types of data, combine it with the health records of individual, and then process that in this secure federated learning methodology so that each individual institution can still retain the privacy of their data, but you can still have AI algorithms really take advantage of it. So if you really, just to summarize, I believe that life science advances will pose unprecedented burden on our cloud infrastructure, and the NI Center is a bright light of hope that will accelerate this research. And we in Intel Labs are really, really excited to collaborate with this team in this journey. With that, I will conclude. Thank you.